As an emergency room doctor for over 15 years, I thought I had seen it all. But Emily Carter's case changed everything I understood about the human brain and its vulnerability to nature's most insidious invaders. The night Emily Carter was wheeled into my emergency room marked the beginning of a case that would rewrite medical textbooks and challenge our understanding of the natural world. Her symptoms defied conventional diagnosis, and the truth behind her condition would prove more terrifying than anything I'd encountered in medical school. The case began like many others in our emergency department. A frantic family member, a patient in distress, the usual rush of nurses and doctors converging to assess the situation. But something about Emily's presentation immediately caught my attention. Her eyes held a strange, glassy quality I'd never seen before, and her movements were erratic in a way that didn't match typical seizure patterns. It started on a cold October night. Emily arrived at Mount Sinai Hospital after a severe seizure, her husband practically carrying her through the emergency room doors. Her initial symptoms seemed routine: headaches, mild confusion, sleep issues, nothing that would raise immediate alarm bells in a busy New York ER. Emily was a respected botanist, her work focused on rare plant specimens from the Amazon rainforest. Her latest acquisition, a peculiar species called Ophiocordyceps novus, sat innocently in her home laboratory. None of us knew then that this plant would become the center of a medical mystery that would challenge modern medicine's understanding of human-plant interaction. Her husband explained how Emily had spent countless hours in her home laboratory, studying the plant's unique characteristics. She'd been particularly fascinated by its response to temperature changes, often working late into the night to document its behavior. The plant itself was unremarkable in appearance, a small grayish-green specimen with delicate tendrils that seemed to shift in the slightest breeze. The plant had been legally acquired through proper channels, with all necessary permits and quarantine procedures followed. Yet somehow, this seemingly innocuous specimen had developed capabilities far beyond its documented characteristics. In the weeks leading up to her admission, Emily had noticed subtle changes in the plant's behavior, increased activity during nighttime hours, and an unusual responsiveness to human presence. During her initial examination, I noticed something odd, a faint, musty smell emanating from her hair. The CT scan results made my blood run cold. Shadow-like formations appeared in her frontal lobe, patterns I'd never encountered in 15 years of emergency medicine. They didn't match typical tumor formations, infection patterns, or any standard neurological condition. Dr. Chen, our resident mycologist, spotted something during the examination that should have been our first warning: microscopic spores clinging to Emily's hair. But we were following standard fungal meningitis protocols, and this observation got buried under the urgent need to stabilize her condition. Looking back, we should have connected the dots sooner. The spores weren't just present in her hair, they had created an almost invisible coating on her skin, so fine it was only visible under certain angles of light. The night staff later reported seeing faint, phosphorescent traces on her pillowcase, but attributed it to hospital laundry detergent residue. The hospital's infectious disease team was baffled by the preliminary test results. Every standard protocol we followed seemed to lead us further from understanding what was happening inside Emily's brain. The spores, when isolated and examined, displayed characteristics never before seen in any known fungal species. They appeared to have evolved sophisticated mechanisms for surviving in the human body, including the ability to mimic human cellular structures. The plant's history became crucial to understanding Emily's condition. Ophiocordyceps novus was discovered deep in the Amazonian rainforest, where local shamans spoke of its unusual properties. The specimen responded to human body heat, releasing spores during the night, a fact we would later discover had sealed Emily's fate. Her deterioration was rapid. Within hours of admission, her cognitive functions began to slip. She struggled to recognize common objects, and her speech became increasingly disorganized. The headaches she had dismissed as stress related now revealed themselves as the first signal of something far more sinister. The security footage from that night shows Emily walking in circles in her room, touching the walls with an almost ritualistic precision. When questioned about these episodes, she had no memory of them. Her husband reported similar behavior at home in the weeks leading up to her admission, finding her standing in their garden at odd hours, hands buried in the soil. The hospital's security cameras captured more than just Emily's nocturnal wanderings. In infrared footage, her body temperature showed unusual patterns, with hot spots concentrating around her head and neck. These thermal anomalies corresponded exactly with the areas where we later discovered the highest concentration of fungal activity. The timing of these temperature fluctuations aligned perfectly with the plant's known nocturnal spore release cycle. The night progressed and Emily's condition worsened. Her temperature spiked to 103 DIF, and her brain activity showed patterns that didn't match any known pathology. The hospital's neurological team gathered, puzzled by the rapid progression and unusual presentation of symptoms. What made this case particularly disturbing was how the infection had masked itself. Initial blood work came back normal. Standard neurological tests showed minimal abnormalities. It was as if something was actively hiding within her system, evading our diagnostic tools with an almost intelligent precision. The lab technicians working on Emily's case noticed something peculiar about her blood samples. Under the microscope, her white blood cells exhibited unusual movement patterns, clustering in formations that resembled mycelia networks. One technician, who had previously worked in mycology, remarked that it looked like the cells were being choreographed by an external force. As dawn approached, Emily began experiencing vivid hallucinations. She spoke of seeing plants growing through the hospital walls, of feeling roots spreading through her mind. At the time, we dismissed these as fever-induced delusions. We would later discover these weren't hallucinations at all, but her brain's interpretation of what was actually happening inside her skull. The samples we took from her cerebrospinal fluid revealed an unprecedented situation. Microscopic filaments, nearly invisible to standard testing, had begun forming intricate networks throughout her central nervous system. These structures bore a striking resemblance to fungal hyphae, but their composition defied conventional classification. They appeared to be synthesizing neurotransmitter-like compounds, effectively hijacking her neural pathways. Her husband brought in her research notes, hoping they might provide some insight. The pages were filled with meticulous observations about the plant's behavior, including detailed sketches of its growth patterns. In the margins of her later entries, the handwriting became increasingly erratic, with strange symbols that seemed to mirror the neural patterns we were observing in her brain scans. The most disturbing discovery came from Emily's home laboratory. The remaining specimens of Ophiocordyceps novus had undergone dramatic changes. The plants had developed unusual growths, their surfaces covered in fine, almost translucent filaments that responded to human proximity. When our team arrived to collect samples, the specimens exhibited synchronized movement patterns, swaying in unison despite the absence of any air current. Our hospital's biosafety protocols were quickly overwhelmed by the complexity of the situation. The spores we collected demonstrated an ability to survive in environments that should have been lethal to any known fungal species. They remained viable after exposure to UV radiation, extreme temperature changes, and even standard sterilization procedures. It was as if they had evolved specifically to overcome human medical defenses. The implications of Emily's case reached far beyond a single patient. If a seemingly harmless plant could develop such sophisticated mechanisms for human infection, what other threats might be lurking in the world's unexplored ecosystems? The boundary between plant and human biology had been breached in a way that would force us to reevaluate our entire understanding of cross-species interaction. As I watched Emily's brain scans that night, I couldn't shake the feeling that we were witnessing something unprecedented in medical history. The infection wasn't just spreading through her brain, it was restructuring it, creating new neural pathways that served some unknown purpose. We were no longer just fighting a disease, we were facing an intelligent adversary that had found a way to bridge the gap between plant and human consciousness. The night shift ended with more questions than answers. Emily's condition had stabilized, but her brain activity continued to show patterns that defied explanation. As I handed over her case to the day team, I couldn't help but wonder if we were prepared for the implications of what we had discovered. The line between human and plant biology had blurred, and we were only beginning to understand the consequences. The true horror of Emily's condition revealed itself through a series of groundbreaking discoveries in our hospital's laboratory. Under the microscope, the spores we found displayed unprecedented behavior. they weren't just surviving in human tissue, they were transforming it. The spores demonstrated an uncanny ability to survive in human tissue temperatures, thriving at precisely 37 degrees Celsius. Initial cultures revealed exponential growth rates that far exceeded any known fungal species. Within hours, the samples had developed complex networks that pulsed with an almost electrical rhythm. The microscopic images revealed thread-like structures weaving through neural tissue with an almost architectural precision. Each new sample showed more extensive colonization, suggesting the infection had been developing long before Emily's symptoms appeared. What disturbed us most was the organized nature of the growth patterns. This wasn't random spread, but a calculated expansion through her neural pathways. The neural tissue samples revealed another unsettling characteristic. The fungal filaments were creating synaptic-like connections with human neurons. These connections weren't random, they followed existing neural pathways, suggesting the fungus could read and replicate human brain architecture. The fungal network grew denser each day, establishing what appeared to be its own neural highways parallel to Emily's natural brain circuits. These spores had evolved an ingenious mechanism of invasion. They bypassed the nasal filtration system by mimicking human protein structures, essentially wearing a biological disguise. Once inside, they traveled along the olfactory nerve, a direct highway to the brain. The spores moved through the cerebrospinal fluid with an efficiency that stunned our research team. Once established in the cerebrospinal fluid, the fungus began producing compounds that altered the brain's chemical balance. The organism wasn't just surviving, it was actively manipulating its host's biochemistry. These changes were subtle at first, manifesting as minor mood alterations and sleep disturbances that could easily be attributed to stress or anxiety. Our standard fungal tests failed spectacularly. The organism had developed a sophisticated protein cloaking mechanism, allowing it to masquerade as white blood cells. Every time we thought we had it cornered, it adapted. It was unlike anything in modern medical literature. The fungus seemed to anticipate our every move, evolving faster than we could develop countermeasures. Each time we introduced a new antifungal agent, the organism would alter its biochemical structure within hours. This level of adaptation shouldn't have been possible in a traditional fungal species. We began to suspect we were dealing with something that had been preparing for human hosts for generations. Our attempts to culture the fungus in standard growth media failed repeatedly. The organism would only thrive in the presence of living neural tissue, suggesting a level of host specificity that defied conventional understanding. When we introduced samples to artificial neural networks in the lab, the fungus immediately oriented itself toward the electrical signals, growing along the same patterns we observed in Emily's brain. Emily's husband, Dr. James Carter, grew increasingly frustrated with our inability to treat her. "You're supposed to be the best hospital in New York," he shouted during one particularly tense meeting. "How can you not know what's happening to my wife?" The ethical debate over experimental treatments reached a boiling point. Traditional antifungal medications proved useless and time was running out. James's medical background only complicated matters. His knowledge of traditional pathology made it difficult for him to accept the unprecedented nature of Emily's infection. During one heated exchange, he demanded we try experimental antifungal treatments that could have caused catastrophic organ failure. We had to balance his desperate need to save his wife with our obligation to first do no harm. During our research, we uncovered a similar case from 2017, a Brazilian farmer who died under mysterious circumstances. The autopsy revealed fungal growth patterns identical to Emily's, but the case never made it to publication. The medical community wasn't ready to accept the possibility of an intelligent fungal invasion. The Brazilian case files contained crucial details that had been overlooked. The farmer's brain tissue samples, preserved in a Sao Paulo laboratory, showed signs of neural reorganization. The fungal colonies hadn't just invaded the tissue, they had restructured it. This matched the changes we were now seeing in Emily's brain scans. The fungus was rewriting her neural architecture according to some alien blueprint. The breakthrough came from an unexpected source. Sarah Chen, a night nurse, noticed something peculiar while checking on Emily. The patient's skin showed a faint luminescence under UV light. This observation led us to discover buried research in botanical journals about bioluminescent properties of certain Amazonian fungi. Further investigation revealed that the luminescence occurred in distinct patterns, particularly during Emily's REM sleep cycles. The timing couldn't be coincidental. The fungus appeared to be most active when her brain entered its most vulnerable state, suggesting it had evolved to exploit natural periods of decreased neural defense. During these episodes, Emily's EEG readings showed unusual synchronization between fungal activity and her brain waves. This bioluminescence wasn't just a curious side effect, it was a sign of active communication between fungal colonies. Under specialized imaging, we observed patterns of light emission that corresponded with neural activity. The fungus had developed its own form of electrochemical signaling, mimicking the very essence of human consciousness. The spores had created an extensive network throughout Emily's central nervous system. They weren't just surviving, they were communicating. Each cluster pulsed with electrical activity that mimicked human neural patterns. The fungus wasn't just attacking her brain, it was attempting to integrate with it. Each cluster operated like a miniature brain, processing information and responding to stimuli. When we applied electrical currents to isolated samples, the fungal networks responded by creating new connections, demonstrating an unsettling ability to learn and adapt. The implications were staggering. We were dealing with a collective intelligence that grew more sophisticated with each passing day. The fungal networks exhibited another disturbing capability. They could store and process information. When exposed to repeated stimuli, the colonies adjusted their growth patterns to create more efficient response pathways. This learning ability suggested we weren't just fighting an infection, we were dealing with an organism capable of building its own neural architecture within a human host. Testing revealed the fungus was producing compounds nearly identical to human neurotransmitters. It wasn't just growing inside her brain, it was learning from it. This discovery sent shockwaves through the medical community. We were witnessing evolution in real time, a fungal organism adapting to human neural architecture. The fungal colonies were synthesizing not just one or two neurotransmitters, but a complex cocktail of neural modulators, serotonin, dopamine, and even novel compounds that resembled human growth factors were being produced in precise quantities. This metabolic feat suggested a deep understanding of human neural chemistry that should have taken millions of years to evolve. Emily's case challenged everything we knew about the blood-brain barrier. The fungus had found ways to penetrate our most sophisticated natural defenses. More disturbing was the possibility that this wasn't a random mutation, but rather an evolved strategy. The plant in Emily's home laboratory had selected her, not the other way around. The fungus's method of breaching the blood-brain barrier revealed sophisticated chemical mimicry. It produced compounds that triggered the barrier's own transport mechanisms, essentially tricking the brain's defensive walls into granting access. This level of biological sophistication suggested years of evolutionary refinement, raising questions about how many failed attempts might have preceded this successful invasion. The spore's protein structures contain sequences never before seen in any known fungal species. They appeared to be custom, designed to interface with human neurons, suggesting a long evolutionary history of human interaction we had somehow missed. This raised disturbing questions about how many other people might have been exposed without anyone noticing. The perfect disguise was just in the fungus's biology, it was in our own blind spots. The protein sequences contained elements that seemed almost engineered, with binding sites perfectly matched to human neural receptors. This level of compatibility raised disturbing questions about the fungus's evolutionary history. Had it been practicing on other mammals before making the leap to human hosts? Or was this the result of a longer, hidden history of human interaction? As we dug deeper, we found more cases, all dismissed or misdiagnosed. The medical community had been blind to an emerging threat, one that had been evolving alongside us, waiting for the perfect moment to cross the species barrier. Emily's neurological deterioration accelerated at an alarming rate. One morning, she looked at her husband with vacant eyes and declared him an imposter. The Capra's delusion had set in, a rare psychiatric condition where patients believed their loved ones had been replaced by identical-looking imposters. But this wasn't just psychological. The fungal invasion had damaged her fusiform gyrus, the part of the brain responsible for facial recognition. The medical team documented her rapid decline with growing concern. Within hours, Emily's speech patterns became erratic, switching between technical botanical terminology and incomprehensible fragments of sentences. Her eyes darted around the room, tracking invisible movements that only she could perceive. The fungal network had begun infiltrating her language centers, creating a disturbing hybrid of scientific knowledge and psychotic delusions. Her husband Mark stood there, frozen, as Emily screamed for security to remove this imposter from her room. The medical team had to physically restrain her as she tried to escape, convinced that everyone around her was part of an elaborate conspiracy. The fungal colonization had spread to her amygdala, the emotional center of the brain, distorting her perception of reality in ways we couldn't have predicted. I watched through the ICU window as she became increasingly agitated. Her brain scans showed massive swelling, forcing us to make the difficult decision to induce a coma. As Emily slipped into unconsciousness, our lab team made a disturbing discovery. Time-lapse footage of the fungal samples showed hyphal growth patterns that seemed almost purposeful, reaching out like fingers searching for something. The induced coma was our only option to prevent catastrophic brain damage, but it also meant surrendering control. As Emily lay motionless, the monitors tracking her neural activity displayed patterns unlike anything in medical literature. Spikes of activity would ripple across her brain in coordinated waves, following the same paths as the fungal growth. The organism wasn't just surviving in her brain, it was orchestrating a complete neural reorganization. Under the microscope, the fungal filaments displayed an unprecedented level of organization. They weren't just growing, they were constructing complex networks that mimicked neural pathways. In the Petri dishes, individual colonies merged and separated in patterns that suggested some form of communication. The implications were staggering. This organism wasn't just surviving, it was learning. The situation in our hospital laboratory became critical. The quarantined plant specimen began releasing spores that attacked other botanical samples. Within hours, healthy plants showed signs of fungal colonization. The organism's ability to adapt and spread through airborne transmission exceeded anything we'd seen before. We had to seal off the entire research wing. The lab technicians noticed something even more unsettling. The infected plants began exhibiting synchronized growth patterns, their leaves turning and twisting in unison despite being in separate containment units. When one sample was exposed to light, others responded simultaneously, suggesting a form of communication we couldn't detect. The fungus had created an invisible network connecting all infected specimens, transforming our sterile lab into its own biological internet. We implemented maximum containment protocols, but the fungus demonstrated an uncanny ability to adapt. It modified its spore size and shape to slip through our finest EPI filters. Even more disturbing, it seemed to respond to our countermeasures, changing its behavior each time we attempted to restrict its spread. The organism was exhibiting a level of strategic response that bordered on intelligence. The lab's air sampling revealed another disturbing development. The fungal spores had begun producing complex organic compounds that mimicked human neurotransmitters. These compounds could potentially cross the blood-brain barrier, suggesting the organism had evolved specifically to interface with human neural tissue. We were witnessing the birth of a biological interface between human and fungal consciousness. During our daily rounds, Dr. Sarah Martinez, a first-year resident, proposed an unconventional treatment intrathecal amphotericin B, delivering antifungal medication directly into the spinal fluid. The suggestion made theoretical sense, but we lacked FDA approval for this delivery method. Time was running out, and bureaucratic red tape was strangling our options. Looking through Emily's medical history, I found something that gave me pause. As a child, she had battled an aggressive form of tuberculosis and survived against all odds. Her immune system had always been remarkably resilient. Now, facing an equally formidable opponent, that same immune system was being systematically dismantled by something far more intelligent than we initially believed. Her childhood battle with tuberculosis had left unique markers in her immune system, creating what we now realized was a perfect environment for fungal colonization. The organism hadn't randomly selected Emily, it had found the ideal host for its evolution. Her enhanced immune response, initially a blessing, had become a catalyst for the fungus's rapid adaptation. Her immune system's unique adaptations had created the perfect storm. The fungus exploited these adaptations, using her enhanced immune response as a blueprint for its own evolution. Each defensive measure her body mounted was studied, decoded, and incorporated into the organism's growing arsenal of biological innovations. It was like watching accelerated evolution in real time, with Emily's immune system inadvertently teaching the fungus how to become more efficient at human infection. The parallels between her childhood illness and current condition were striking. Both infections had targeted her nervous system, but while tuberculosis had attacked indiscriminately, this fungus showed precise targeting. Her previous medical records revealed patterns we hadn't noticed before. Her immune system had unique characteristics that might have made her an ideal host for this evolutionary experiment. The fungal invasion progressed with frightening precision. Each new scan revealed more extensive infiltration of her neural pathways. The organism wasn't just spreading randomly, it was following a pattern, targeting specific regions of her brain in a predetermined sequence. We were watching an orchestrated takeover of human neural architecture. Each affected region corresponded to a specific cognitive function, memory, emotion, motor control. The fungus wasn't just colonizing her brain, it was mapping it, learning its architecture. In rare moments of consciousness, Emily exhibited behaviors that seemed almost choreographed, as if something else was testing the limits of her neural circuitry. During our observations, we noticed periods of intense neural activity coinciding with the fungal network's expansion. These bursts matched patterns typically seen during deep learning processes, suggesting the organism was not just colonizing but studying its host. Emily's brain had become both a battlefield and a classroom for this evolutionary breakthrough. The fungal network's learning capability exceeded our worst fears. It demonstrated an ability to anticipate and counter our treatment attempts before we even administered them. Traditional antifungal medications were rendered useless as the organism adapted its cellular structure to resist their effects. We were no longer fighting a simple infection, we were engaged in a biological arms race against an opponent that could evolve faster than we could develop new treatments. The pressure inside Emily's skull continued to rise despite our best efforts. Her brain tissue showed signs of reorganization, with new neural pathways forming in patterns that matched the fungal growth. It was as if the organism was building its own network within her mind, using her neurons as a template for something entirely new. The new neural pathways defied conventional medical understanding. They carried signals faster than normal neurons, creating shortcuts through her brain that shouldn't have been possible. When we monitored her brain activity, we detected patterns that resembled both human consciousness and something entirely alien. It was as if two distinct forms of intelligence were competing for control of her cognitive processes. The hybrid neural pathways created by the fungus demonstrated capabilities that defied our understanding of cellular biology. They could transmit signals using both electrical and chemical mechanisms simultaneously, achieving computational speeds that exceeded normal human neural processing. In essence, the fungus was upgrading Emily's brain, but for what purpose remained unclear. These enhanced pathways seemed to be building towards something, a new form of consciousness that was neither fully human nor entirely fungal. As we prepared for emergency intervention, I couldn't shake the feeling that we were dealing with something beyond our current understanding of biology. This wasn't just an infection, it was an invasion by an organism that had potentially been planning this evolutionary leap for millions of years. The operating room fell silent as we prepared for Emily's brain biopsy. The surgical team moved with practiced precision, but there was an undercurrent of tension. This wasn't a standard procedure. We were facing an organism that had already demonstrated remarkable adaptability. The scrub nurse handed me the specialized biopsy needle, its titanium shaft gleaming under the harsh operating room lights. We had modified the standard equipment to account for the unique nature of our target. The fungal filaments had shown unusual resistance to metal in our preliminary tests, corroding standard surgical steel within hours of contact. The nurses had meticulously prepared the surgical field, their movements betraying the gravity of the situation. Emily lay unconscious, her head secured in the stereotactic frame. The pre-operative MRI scans displayed on the monitors showed dark patches spreading through her brain tissue like ink in water. Dr. Chen, who had performed countless biopsies over his 20-year career, took a moment to study these images one final time. As Dr. Chen made the initial incision, I monitored the vital signs. The moment we accessed the basal ganglia, everyone in the room drew a collective breath. Silvery filaments, clearly visible to the naked eye, had woven themselves through the tissue. They pulsed with an almost electrical rhythm, suggesting some form of communication network. The surgical team worked methodically, extracting multiple samples from different regions of Emily's brain. Each new specimen revealed more extensive colonization than the last. The fungal network had established itself most prominently in areas controlling emotion and memory. The limbic system showed particularly dense growth patterns. This targeting suggested an unsettling level of specificity in the organism's behavior. The tissue sample revealed an architectural complexity that defied conventional understanding. Under the microscope, the fungal structures displayed patterns reminiscent of neural networks, but with an alien geometry that made even veteran pathologists uncomfortable. The filaments seemed to pulse with a faint bioluminescence, visible only in the darkened laboratory. In the lab, the genomic sequencing results confirmed our worst fears. The fungal DNA matched perfectly with samples taken from Emily's plant. We had documented the first case of florogenic brain colonization in human history. The implications were staggering. A common houseplant had evolved the ability to hijack human neural pathways. The sequencing data revealed something even more disturbing. The fungus had incorporated fragments of human DNA into its genome, creating hybrid sequences that allowed it to produce human-like proteins. This explained its ability to evade the immune system and interface with neural tissue. The organism had effectively created a biological bridge between kingdoms, defying our fundamental understanding of species barriers. The genetic modifications we observed went beyond simple adaptation. The fungus had developed specialized structures that mimicked human astrocytes, the support cells crucial for neural function. These hybrid cells could process and transmit information using a combination of chemical signals from both species. In essence, the organism had created its own biological translation system between plant and human cellular communication. The team worked through the night, analyzing every aspect of the fungal invasion. The organism had developed specialized structures that interfaced directly with human neurons, creating hybrid synapses that shouldn't have been possible. Each new discovery raised more questions about the evolutionary leap this organism had made. The ethical debate erupted immediately. Dr. Harrison from the research department argued passionately for preserving the plant specimen. This could revolutionize our understanding of cross-species neural networks, he insisted. But I couldn't forget Emily's vacant stare, her personality slowly being erased by this invasion. The hospital board convened an emergency meeting to address the situation. Legal experts warned about potential biohazard regulations, while research institutions worldwide began requesting samples. In the midst of this bureaucratic storm, Emily's husband sat alone in the waiting room, clutching a photo of their wedding day, when she had carried a bouquet of her beloved exotic flowers. Then something unexpected happened. Emily's vital signs began to stabilize. Further analysis revealed the fungus was producing serotonin analogs, chemicals nearly identical to human neurotransmitters. Was this a form of symbiosis or something more sinister? The organism appeared to be maintaining its host's basic functions while restructuring her neural architecture. The neurotransmitter production wasn't random. Analysis revealed the fungus was synthesizing compounds that targeted specific neural pathways, those involved in decision-making and emotional regulation. The chemical profile matched that found in the plant's natural defense system, but had been modified to interact with human receptors. This adaptation suggested the organism was actively attempting to influence its host behavior. The fungal network's production of neurotransmitters followed a precise circadian rhythm, peaking during the hours Emily would normally be asleep. This temporal pattern matched the natural cycle of the plant in her home, suggesting a sophisticated biological synchronization. The organism wasn't just surviving in her brain, it was maintaining its original daily cycles while adapting to its new host. The medical team documented every change in Emily's condition. Her brainwave patterns showed unusual synchronization with the fungal growth cycles. During certain periods, her EEG readings matched the electrical activity measured in the plant specimen, suggesting a form of cross-species neural entrainment never before observed in medical literature. The CDC response was swift and severe. Within hours of notification, a team of officials arrived in biohazard suits. They implemented protocols typically reserved for pandemic scenarios. The entire floor was quarantined, and every staff member who had contact with Emily or the plant was screened for spore exposure. The quarantine procedures transformed the hospital wing into something resembling a military installation. Air filtration systems were upgraded and UV sterilization units were installed in every corridor. The CDC team established a mobile laboratory complete with electron microscopes and genetic sequencing equipment. Their presence drew media attention, forcing the hospital administration to issue carefully worded statements that revealed nothing while attempting to prevent panic. The quarantine procedures revealed another concerning aspect of the infection. Environmental sampling detected trace amounts of fungal spores in the ventilation system, but only in areas Emily had frequented. The spores remained dormant until exposed to human body temperature, suggesting they were specifically engineered to target human hosts. This discovery led to a complete overhaul of the hospital's air handling systems and the implementation of thermal decontamination protocols. Each new test revealed layers of complexity we hadn't anticipated. The fungal cells had developed specialized structures resembling myelin sheaths, the protective coating around human nerve fibers. This adaptation allowed them to transmit electrical signals at speeds approaching those of natural neurons. In effect, the organism had engineered its own version of human neural architecture. The lab team discovered trace amounts of novel compounds in Emily's cerebrospinal fluid. These molecules shared structural similarities with both plant hormones and human growth factors, suggesting the fungus was actively directing tissue development. It had created a hybrid biochemical environment that served its own mysterious purposes. I watched through the observation window as they collected samples. The fungal filaments had created an intricate network throughout Emily's brain tissue, following patterns that seemed almost architectural in their precision. This wasn't random growth, it suggested a level of intelligence we hadn't believed possible in a fungal organism. The fungal network's architecture challenged everything we knew about biological information processing. It had created redundant pathways throughout Emily's brain, establishing what appeared to be backup systems for vital functions. This level of sophistication suggested the organism had evolved specifically to integrate with human neural tissue. The question wasn't just how this happened, but why this particular plant had developed such capabilities. The implications of our discovery sent shockwaves through the medical community. If a common houseplant could evolve this level of neural manipulation, what other threats might be lurking in our homes? The boundary between plant and human biology had been breached, and we were only beginning to understand the consequences. The neural hijacking we observed in Emily's case was unprecedented. Through advanced imaging, we watched in horror as the fungal hyphae systematically rewired her neural pathways. The process mirrored the infamous Ophiocordyceps unilateralis, the zombie ant fungus, but with frightening sophistication. Unlike its ant-controlling cousin, this strain had evolved to manipulate the most complex organ in the known universe, the human brain. The fungal infiltration moved with calculated precision through Emily's neural architecture. Within days, the hypha had established complex networks that pulsed with electrical activity, mimicking the rhythm of normal brain waves. Traditional antifungal treatments proved useless against this biological invasion. The fungus had developed mechanisms to shield itself using Emily's own neural tissue as a protective barrier. The fungal filaments created intricate networks that resembled neural maps, but with a disturbing difference. Each connection served the fungus's goal of domination. Under the microscope, we observed the hypha secreting neurotransmitter like compounds that mimicked human brain chemistry. These compounds didn't just interfere with normal brain function, they enhanced certain neural pathways while suppressing others. The fungus was essentially building a secondary nervous system within Emily's brain, one that could override her own neural commands. Our team worked around the clock to develop a treatment strategy. We combined cutting-edge cancer immunotherapy with modified antiretroviral drugs, hoping to target the fungus without destroying Emily's neural tissue. Time wasn't on our side. The fungal network had begun sporulating inside her skull, triggering a devastating countdown. If we couldn't stop the reproductive cycle within 72 hours, the damage would become irreversible. The pressure in the ICU was palpable. Every hour brought new challenges as Emily's brain chemistry fluctuated wildly. The fungus seemed to anticipate our interventions, adapting faster than we could respond. It was like playing chess against an opponent who could reshape the board. Each scan revealed new horrors. The fungal network had established control centers in key regions of Emily's brain, particularly in areas controlling basic survival functions. It wasn't just growing randomly, it was targeting specific neural clusters with surgical precision. The hippocampus, responsible for memory formation, showed extensive colonization. This explained why Emily's recent memories were becoming increasingly fragmented, while older ones remained intact. The fungus was actively editing her consciousness, perhaps ensuring she wouldn't remember enough to fight back effectively. The fungal colonization didn't stop at neural control. Blood samples revealed elevated levels of novel compounds, molecules that shouldn't exist in human biology. The fungus was manufacturing its own neurotransmitters, creating a chemical soup that kept Emily's vital organs functioning while it rewired her brain. In a twisted way, it was keeping her alive to serve its own purposes. During a routine check, Dr. Rivera noticed something alarming on the environmental monitors. The ambient temperature in Emily's room had risen by 2 degrees, matching the optimal conditions for fungal reproduction. The organism was actively modifying its microenvironment, demonstrating environmental engineering capabilities that should have been impossible for a fungus. Then came the breakthrough we never expected. During a brief period of consciousness, Emily began frantically scribbling on her notepad. At first, we dismissed the drawings as nonsensical, abstract patterns that resembled tree branches or river systems. But Dr. Zhang, our resident neurologist, recognized something crucial. The drawings matched the exact growth patterns of the fungal networks in her brain. Emily wasn't just fighting the infection, she was mapping it. Emily's drawings revealed something else, a pattern of fungal growth that followed the brain's natural electrical impulses. The organism wasn't just spreading randomly, it was using her neural activity as a map, following the paths of least resistance through her brain tissue. This explained why certain regions were more heavily affected than others. The fungus had essentially created a biological GPS system, using Emily's own brain activity to guide its expansion. We discovered another disturbing aspect of the infection. The fungal network had developed specialized structures that could detect and respond to electromagnetic fields, essentially creating a biological antenna system within Emily's brain. These structures allowed the fungus to sense and react to changes in its environment with almost instantaneous precision. It could adjust its growth patterns and chemical production based on external stimuli, displaying a level of environmental awareness that bordered on consciousness. The climate change connection became impossible to ignore. Rising global temperatures had created perfect conditions for fungal evolution. Species that had never posed a threat to humans were adapting at unprecedented rates, developing resistance to our body's natural defenses. Emily's case wasn't just an anomaly, it was a warning. The fungal reproduction cycle accelerated. Microscopic examination revealed spores developing protective coatings that could resist conventional antifungal treatments. We were watching evolution in real time, driven by an intelligence that defied our understanding of plant biology. The spores themselves showed signs of unprecedented sophistication. Their outer shells contained compounds similar to those found in human immune cells, allowing them to move through the bloodstream undetected. More disturbing still, they could alter their chemical signatures in response to our treatments, making each new generation more resistant than the last. The fungus wasn't just reproducing, it was evolving in real time, using Emily's immune system as a training ground for its offspring. The fungal spores showed signs of quantum coherence, a phenomenon previously unknown in biological systems of this scale. This explained their ability to coordinate actions across vast networks of neural tissue. Each spore functioned as part of a larger quantum computer, sharing information instantaneously across the entire network. The fungus had evolved beyond simple biological processes into something that challenged our understanding of life itself. In a moment of horrifying clarity, we realized the fungus wasn't just surviving, it was learning. Each failed treatment attempt seemed to make it stronger, more resilient. The network in Emily's brain had become a living laboratory for rapid adaptation, using her neural pathways to process and respond to threats. Deep in Emily's temporal lobe, we discovered something that defied explanation. The fungal network had created structures that mimicked synaptic junctions, but with a crucial difference: they could transmit signals faster than human neurons. These super synapses formed a parallel processing system, like a biological computer running alongside Emily's natural brain functions. The fungus wasn't just parasitizing her brain, it was upgrading it for its own purposes. The super synapses created by the fungus exhibited another unprecedented characteristic. They could store and process information in ways that resembled computer memory systems. Each fungal node acted as a biological data storage unit, archiving information about immune responses, treatment attempts, and environmental conditions. This distributed intelligence allowed the fungus to learn from every interaction, becoming more sophisticated with each passing hour. We weren't just fighting an infection, we were battling an emerging form of biological artificial intelligence. As the 72-hour deadline approached, Emily's vital signs began to fluctuate wildly. The fungal network was preparing for its final reproductive surge, and we were running out of options. In her brief moments of lucidity, Emily's eyes reflected a terrifying awareness. She could feel the organism thinking through her neural pathways, using her brain's processing power for its own evolution. Desperation drove innovation. In the operating room, we prepared for a procedure that had never been attempted. Using focused ultrasound technology, we planned to create controlled pathways, fungal highways, that would redirect the organism's growth toward removable synthetic membranes. The risks were enormous, but inaction meant certain death. The concept had emerged from a late-night discussion between Dr. Chen and Dr. Rodriguez, inspired by their research on neuroplasticity. If fungal growth followed electromagnetic pathways, they reasoned, controlled ultrasound could essentially create artificial roads for the infection to travel. The theory was sound, but the stakes were unprecedented. One miscalculation could cause catastrophic damage to Emily's neural architecture. The procedure room hummed with tension as we prepared Emily. Each team member understood their role in this unprecedented intervention. The ultrasound equipment, normally used for tumor ablation, had been modified specifically for this purpose. Our bioengineers had worked through the night to calibrate the frequencies needed to influence fungal growth patterns without damaging vital brain tissue. The surgical team moved with precise coordination. Dr. Shen, our ultrasound specialist, carefully mapped the fungal networks illuminated by our advanced imaging. Each pulse of focused energy carved microscopic channels through Emily's brain tissue, creating paths of least resistance for the fungal hypha to follow. The initial ultrasound pulses revealed something unexpected. The fungal networks showed signs of organized behavior. They weren't growing randomly, but following specific neural pathways, particularly those rich in serotonin receptors. This discovery forced us to adjust our approach mid-procedure. Instead of creating straight channels, we had to design curved pathways that mimicked the brain's natural architecture. The discovery of this organized behavior sent shockwaves through the medical team. The fungus wasn't just surviving, it was adapting, learning, and possibly even communicating with Emily's neural networks. Dr. Chen's hands trembled slightly as she adjusted the ultrasound parameters. Each pulse had to be perfect, each pathway precisely calculated to exploit the organism's apparent intelligence. While we worked, our pharmaceutical team fought bureaucratic battles. The FDA emergency approval process typically took months, but Emily had hours. Through a series of urgent conference calls, we secured permission to use an experimental cross-species antifungal compound developed using data from Emily's original plant specimen. The compound itself was a marvel of biochemistry. Based on a modified polyene structure, it could bind to fungal cell membranes while leaving human neurons intact. Laboratory tests showed it could effectively redirect fungal growth without triggering the organism's defensive responses. But we had never tested it in a living human brain. The plant itself became our unexpected ally in its final moments. During a desperate attempt to extract viable spores for research, the specimen underwent a catastrophic collapse. But as it died, it released a complex chemical cascade, a last gasp that proved to be our salvation. The compounds, when analyzed, revealed a natural fungal inhibitor that the plant had evolved as a defense mechanism. Initial spectroscopy revealed compounds never before documented in botanical literature. The plant's final moments unleashed a biochemical treasure trove, molecules that seemed to bridge the gap between plant and human biology. In the lab adjacent to the operating room, biochemists worked frantically to isolate and synthesize these compounds before they degraded. The plant's death triggered a cascade of events that none of us could have predicted. As its cellular structure broke down, it released not just inhibitors, but also complex sugars and proteins, molecules that seemed almost designed to interface with human neural tissue. Later analysis would reveal that the plant had developed these compounds through centuries of evolution, possibly as part of a larger survival strategy. We synthesized the compound within hours. The timing was critical. Emily's intracranial pressure was reaching dangerous levels. As we administered the treatment, the monitors showed an immediate response. The fungal networks began to retract, following our engineered pathways toward the collection membranes. Time became our enemy. Each hour brought new challenges as Emily's body reacted to both the treatment and the fungal die-off. We monitored her brain activity constantly, watching for signs of seizure or tissue damage. The fungal mass began moving through our engineered channels, but slower than we had hoped. We could only watch and wait, knowing that any attempt to accelerate the process could prove fatal. The next 24 hours tested every limit of modern medicine. We watched as the fungal mass gradually migrated along our created channels, leaving behind what we called ghost networks, preserved neural pathways that mapped the organism's former presence. These ethereal traces would later provide invaluable data about how the fungus had integrated with Emily's neural architecture. These ghost networks revealed something extraordinary. The fungus had created neural pathways that shouldn't have been possible. In some areas, it had established connections between brain regions that normally didn't communicate directly. Some of these novel connections appeared to enhance cognitive processing in unexpected ways. Emily's brain had been essentially rewired, though whether this was intentional or a side effect of the infection remained unclear. The victory came at a cost. The plant's death released its entire chemical arsenal, not just the compounds we needed. Some of these substances were highly toxic, forcing us to balance treatment with aggressive filtration of Emily's blood. But somehow, remarkably, the combination of focused ultrasound guidance and the plant's chemical legacy worked. The fungal infection began to recede. The procedure established a new paradigm in neurosurgical intervention. By combining ultrasound technology with biological guidance systems, we had essentially created a method for shepherding invasive organisms out of the brain. This technique would later prove valuable in treating other types of neural infections, though none quite as dramatic as Emily's case. The implications extended beyond medicine into the realm of human evolution. The fungus had demonstrated an ability to interface with human consciousness in ways that challenged our understanding of intelligence. It raised profound questions about the nature of cognition and the potential for inter-species neural communication. Some researchers even suggested that similar fungal interactions might have played a role in human brain evolution, though such theories remained highly controversial. In the aftermath, the World Health Organization moved with unprecedented speed. New guidelines for exotic plant ownership were drafted within weeks. Emily's case had exposed a vulnerability in our species that no one had anticipated. Our brains could be colonized by evolved plant organisms. The medical community struggled to process the implications. We had witnessed not just an infection, but a form of biological intelligence that could hijack human consciousness. The boundaries between plant and human neurology had been blurred in ways that would reshape our understanding of both. Further investigation revealed that the fungus had created novel synaptic connections during its occupation of Emily's brain. These connections somehow enhanced certain cognitive functions while suppressing others. It raised disturbing questions about the nature of consciousness and the potential for biological manipulation of human thought processes. As Emily stabilized, our success drew international attention. Research facilities worldwide began screening their botanical collections for similar fungal adaptations. The incident had opened a new frontier in medical science, one where the line between healing and horror balanced on a microscopic edge. Recovery proved to be a different kind of battle. As Emily's primary physician, I watched her navigate the challenging months of rehabilitation with the same determination that had kept her alive. The fungal invasion had left its mark, not just in the ghost networks visible on our scans, but in the way her mind now processed information. The neural pathways affected by the fungal invasion had created unexpected challenges. Simple tasks like writing her name or recognizing familiar faces would sometimes elude her, while complex botanical calculations came to her with supernatural ease. The medical team documented several instances where Emily could recite entire genetic sequences of plants she had never studied, as if the fungal network had left behind a vast botanical database in her mind. The physical therapy sessions were particularly challenging. Emily's motor functions had been compromised in subtle ways. Her fingers would sometimes twitch uncontrollably when handling certain plants, as if responding to invisible stimuli. The medical team documented each anomaly, building a comprehensive picture of how the fungal invasion had rewired her nervous system. Her husband Mark attended every session, his presence anchoring her to reality when the phantom sensations became overwhelming. During our initial recovery sessions, Emily struggled with memory gaps that appeared random at first but revealed a pattern. She could recall complex botanical facts with perfect clarity, yet sometimes forgot her husband's face. More disturbing were the phantom plant hallucinations, vivid sensations of roots growing beneath her skin that would wake her screaming in the night. These episodes led us to discover something unprecedented in medical literature. Emily's brain had developed new neural pathways that seemed to process plant biochemistry in ways we couldn't explain. During one session, she correctly identified a toxic plant specimen without any visual cues, solely through what she described as molecular whispers. We later confirmed the plant was indeed emitting trace amounts of toxic compounds, but at levels our instruments barely detected. This heightened sensitivity extended beyond just toxic plants. During her recovery, Emily demonstrated an uncanny ability to predict when plants were about to flower, even when kept in sealed rooms without natural light cycles. Her brain appeared to process plant biochemistry on a fundamental level, interpreting signals that our most sophisticated equipment couldn't detect. The medical team began documenting these predictions, finding her accuracy rate exceeded 99%. The scientific community didn't wait for Emily's full recovery to begin studying her case. Her experience sparked a groundbreaking paper in Nature that revealed how certain fungi could produce compounds nearly identical to human neurotransmitters. The implications revolutionized our understanding of plant-human interactions at a molecular level. What haunted me most were the unanswered questions. During genetic analysis, we discovered Emily carried a rare mutation affecting her potassium channels, a variation present in less than 0.1% of the population. This mutation, we theorized, had made her brain tissue irresistible to the fungal spores. It was as if the plant had recognized her as an ideal host, raising disturbing questions about the intelligence behind its evolution. The mutation wasn't just a genetic anomaly, it represented a potential evolutionary bridge between plant and human biology. Our research team identified similar genetic markers in historical cases of unexplained plant-related incidents, suggesting Emily wasn't the first to experience this type of cross-kingdom interaction. The implications sent shockwaves through the botanical research community. Further genetic research revealed clusters of similar cases throughout history. A 19th century botanist in India who could allegedly communicate with sacred trees. A Brazilian shaman who predicted plant behavior with impossible accuracy. A Japanese gardener who survived ingesting deadly nightshade without ill effects. All carried variations of Emily's unique genetic marker. These cases had been dismissed as folklore, but Emily's experience forced us to reevaluate these historical accounts through a scientific lens. Emily's rehabilitation included an unexpected form of therapy. In a secure greenhouse facility, under strict biosafety protocols, she began working with plants again. It was both treatment and research, each interaction carefully monitored for signs of unusual plant behavior. The courage this took impressed everyone on the medical team. The most remarkable aspect of Emily's recovery was her resilience. Despite everything she'd endured, she emerged as a powerful advocate for fungal research funding. Her testimony before congressional committees helped establish new protocols for studying potentially dangerous botanical specimens. She turned her nightmare into a mission to protect others. Her advocacy work revealed darker truths about the botanical research world. Several private corporations had been experimenting with similar fungal species, seeking to harness their neural manipulation capabilities. Emily's testimony led to the shutdown of 3 unauthorized research facilities and the establishment of the International Botanical Safety Commission. 6 months after her ordeal, I joined Emily for her final brain scan. The ghost networks had begun to fade, but they'd left behind something unexpected, new neural pathways that seemed to enhance certain cognitive functions. She could now perceive subtle changes in plant behavior that others missed, as if her brain had retained some evolutionary advantage from its fungal invader. The enhanced neural pathways manifested in unexpected ways. Emily developed what she called green synesthesia, the ability to perceive plant emotions as colors and patterns in her visual field. Under controlled conditions, she could accurately identify plant stress, growth patterns, and even chemical composition simply by looking at a leaf. Independent verification confirmed her readings matched laboratory analysis with startling precision. As I write this final entry in her case file, I'm struck by the profound implications of what we witnessed. Emily's experience forced us to confront an uncomfortable truth: the boundary between human consciousness and other forms of biological intelligence may be more permeable than we ever imagined. The medical community remains divided on the long-term implications of Emily's case. Some researchers argue her enhanced plant perception represents a forced evolutionary leap, while others warn it could be the precursor to a new type of biological warfare. The Department of Defense has quietly established a task force to monitor similar cases worldwide, though none have matched the complexity of Emily's experience. Today, Emily tends her secure greenhouse with a mixture of caution and fascination. She's helped design new safety protocols that have become the global standard for botanical research facilities. But sometimes, when she thinks no one is watching, I see her studying her plants with an almost knowing expression, as if she's learned to read their hidden intentions. The secure greenhouse became a living laboratory for studying plant-human interaction. Emily's presence seemed to affect plant growth patterns in measurable ways. Specimens under her care demonstrated accelerated growth rates and unusual adaptive behaviors. More intriguingly, these plants showed heightened resistance to pathogens, as if they had absorbed some of her enhanced immune response. The implications for agricultural science were staggering. Her work has inspired a new field of study in neurobotany, examining the potential for plant-human neural interfaces. While most researchers approach this field with caution, Emily's unique perspective has opened doors to understanding how plant intelligence might be safely studied and even utilized in medical treatments. The fungal networks that once threatened her life have become a blueprint for developing new therapeutic approaches to neurological disorders. The medical applications of Emily's case expanded beyond neurology. Her enhanced plant perception led to the identification of several novel compounds with therapeutic potential. A previously unknown fungal metabolite, isolated from samples taken during her infection, showed promise in treating neurodegenerative diseases. The fungal networks that nearly claimed her life became templates for developing new drug delivery systems that could cross the blood-brain barrier with unprecedented precision. Her case changed how we view the natural world. In every seed, every spore, every seemingly innocent leaf, there might lie the potential for both miracle and menace. The question Emily now asks her students is both warning and wonder. When you bring a plant into your home, do you ever wonder if it's dreaming of you?